Today in this video, I'm going to discuss the maturation of the endosome, the specific processes that these are involved in, and the resulting, of course, lysosome or the endolysosome. To begin with an overview of my schematic, we have a plasma membrane, we have transmembrane receptors, we have red to indicate the cargo as of now, and then we have specific phospholipids, but specifically of interest in this discussion is going to be phosphatidylinositol. And we recall from our previous studies that phosphatidylinositol is a common phospholipid found in biological membranes, but it has a specific sugar group that can be phosphorylated at specific regions, which is crucial for the specific processes that we will discuss. To begin with the actual process, we have PI kinases that actually phosphorylate these phosphatidylinositol. Okay, so PI kinase, recall that kinases add phosphate groups, phosphatases remove those groups. So we have a PI kinase that modifies this phosphatidylinositol, adding these two phosphate groups. We then have a given adapter protein. We have AP2 that binds phosphatidylinositol. This is actually going to cause a conformational change in the adapter protein, so AP2. It's then going to bind the respective phosphate regions in addition to the actual transmembrane cytosolic exposed regions, forming AP2 receptor PIP complex. Recall that PIP just indicates phosphatidylinositol phosphate. So that's where that last P comes from. For step number 3, we have the actual formation of a clathrin-coat, which this itself is actually responsible for the formation of the budding vesicle. So we have on the outside region the extracellular component, so outside the cell, and then the actual cytosolic component, which that is where the budding is actually going to occur. As depicted in my illustration, we have the formation of a coated pit. Okay, the coat is composed of clathrin, and the clathrin is able to associate with this specific complex due to a positive feedback loop, okay, and it's self-amplifying. All right, so as we have AP2 binding, it is then going to recruit itself as well as clathrin, amplifying this process even further. So as clathrin binds, it is likewise going to stimulate the association of additional adapter proteins. As also depicted in the illustration, we have membrane bending proteins such as BAR and dynamin, which allow the constriction, okay? And this is going to be accomplished by, say, recruiting, changing the lipid composition at the actual membrane. So that results in the formation of an endocytosed vesicle. And this endocytosed vesicle is comprised of, say, all of these membrane proteins, so the transmembrane receptors, as well as other proteins, in addition to, say, just phospholipids and other endocytosed contents. These contents could be anything that just happen to be in the proximity of that specific endocytosed region. Okay, as further depicted, we have endocytosed vesicles, plural. So these vesicles are going to fuse with each other, giving rise to an early endosome. Okay, and allow us to notice that these early endosomes have a lot of these tubule projections, okay? And these tubule projections are actually critical for these processes that we will subsequently discuss. And these are characteristic of early endosomes. As we observe later on in the maturation process, we actually lose the tubular extensions, okay? To move on, we are going to discuss step number 5, which is vesicular transport, and that just refers to the different routes that these actual vesicles and their contents can take upon budding from the actual early endosome. So as we observe here, we can have cycling that occurs, so actual budding from the early endosome. Notice that these buds actually arise from the tubule projections, okay? Okay, and recycling, as the name implies, it is the membrane proteins, the original membrane proteins that were endocytosed into the cell, being translocated from either the early endosome directly to the original cytosolic phase, so where it came from in that membrane. Okay, and recycling can likewise result from the transport of contents of vesicle budding off of the early endosome from, once again, the tubule projection to the recycling endosome before making its way back into the original membrane. So once again, this is recycling. Okay, and a second route these vesicles could actually take is via transcytosis, which is essentially just a fancy term for saying taking contents from one cytosolic region and translocating them to a different location. So for example, I'm going to endocytose contents from above me, all right? I'm going to take these in. I'm going to function, say, my body as the early endosome. Then these contents are going to bud off of my main body and be transported to, say, somewhere over here. Okay, so it's taking contents from one cytosolic phase and translocating them to another cytosolic region. So that is transcytosis. Okay, and to discuss a third type of vesicular transport, we have TGN Exchange, which is trans-Golgi network exchange. The endosome, however, is mostly going to be on, say, the receiving end from what we know, and it is going to be receiving hydrolases and V-type ATPases. More on this later. So real quickly to discuss the Golgi apparatus, we know the Golgi apparatus to be composed of the cis-Golgi network, so CGN and the TGN. However, I did not depict the CGN. The CGN is the receiving end, and the TGN is the transporting end. So the CGN receives contents, it receives proteins. We recall that proteins are co-translationally injected into the endoplasmic reticulum. These are then exported via vesicles to the CGN. The CGN modifies proteins, and then these proteins are going to be exported from the TGN. These contents, say lysosomal hydrolases, after receiving their phosphate group, can be transported and inserted into these endosomes. And we know that this TGN exchange process occurs throughout the entire life cycle, the entire maturation process of a given endosome. So we could expect that the hydrolases and the V-type ATPases are going to be most concentrated in, say, the endosome or lysosome, which is the final step of the maturation process. Back to the actual maturation process of the early endosome. Over time, the actual early endosome loses its tubule extensions and it undergoes vacuolar changes. So what I am essentially saying is that we are losing these tubule extensions and are therefore losing the capacity to transcytose and recycle, which is, yes, what we'd expect, because these late endosomes contain contents that are going to be subsequently degraded by lysosomal hydrolases in the lysosome. Okay, and we also observe vacuolar changes, which are essentially changes in the region with the greatest volume. Okay, we recall that the vacuolar space has the greatest volume, but the tubule extensions have the greatest surface area, which is most suitable for the actual formation of buds and these vesicles that could be actually recycled and transcytosed. But once again, if we lack these extensions, we can no longer transcytose and recycle these contents, so all of these contents will be degraded. We also observe a change in the Rab proteins, okay, going from Rab5 to Rab7. We know that Rab proteins are associated with specific Rab effectors, organelle changes, and the actual location of the specific organelles. The exact mechanisms underlying Rab proteins are unknown, but we know that Rab proteins, say Rab5, as soon as it manifests in, say, a given early endosome, it has a positive feedback loop to increase the concentration of, say, Rab7, while a negative feedback loop on itself. So we observe a natural transition, a natural maturation process from Rab5 to Rab7. So over time, as we would expect, we have a high concentration of Rab5 up here in the early endosome and a high concentration of Rab7, but a low concentration of Rab5 in the late endosome. I should also mention that specific bacteria can actually alter these Rab proteins to change the fate of that specific vesicle that can result in it not being degraded in a lysosome, so it not being killed. Though that is observed during phagocytosis via, say, macrophages, so that is not of our concern here. So I will move on. Next, we observe actual changes in the location of these early endosomes, likely facilitated by the changes in the concentrations of Rab proteins. We have movement toward the nucleus from the peripheral cell membrane region. So this movement toward the nucleus is likely an additional result of the Rab cascade. Okay, so the transition from Rab5 to Rab7. Finally, we also observe the formation of intraluminal vesicles. And these are functionally important because we recall that these membrane proteins up here that weren't recycled are still present. And what that means is these proteins, these membrane receptors, are still in the actual membrane of the endosome and are still thus functional and can therefore still bind its ligand. So we sequester these transmembrane receptors in intraluminal vesicles to prevent, say, unwanted receptor ligand interactions, to prevent, say, unwanted processes from being initiated. And while I am discussing the formation of these intraluminal vesicles, I should also mention that receptor ligand fate depends on signal sequences and acidity. So to reference the K-del receptor, we know that changes in acidity result in different interactions with a specific receptor and a ligand. So if we recall from biochemistry and with our Km values and the interaction between a receptor and a ligand, the interactions change depending on the pH. Because we recall that at different pHs, amino acids exist in different protonated, deprotonated states, and this thereby influences receptor ligand affinity. And this allows us to realize that at different pHs, which is maintained in different organelles, the receptor and the ligand have a different affinity. And what this means is that a receptor and a ligand can either be bound inside the different organelle or dissociated. So, for example, depending on the receptor ligand affinity in, say, an acidic environment, if the affinity between the receptor and the ligand is strong, though the receptor is recycled, those two contents together in its bound state will be recycled. However, if it is a weak affinity, but the receptor is recycled, the ligand in the acidic pH is going to be dissociated and the receptor will be recycled to the membrane. However, the ligand, because their interaction is weak at a low pH, only the ligand, only the thing that binds to the receptor is going to actually be degraded. But the actual receptor itself is going to be back in a membrane. And that is very common, say with LDL receptors binding ApoB particles, such as an LDL particle. And the signal sequence is responsible for dictating whether or not the receptor is going to be either recycled, transcytosed, or degraded. If we have no signal sequence, it is going to follow the endosomal pathway and be degraded. And this is why, say, any specific debris that is foreign is going to be degraded because it doesn't have a specific, say, signal sequence that corresponds to our machinery. Next, we discuss multivesicular bodies, aka late endosomes. These are synonymous with each other. The term multivesicular body was just derived based on how a late endosome looks under a microscope. We notice that we've completely lost our tubular extensions, so we wouldn't observe any transcytosis nor recycling, and only subsequent fusion with an endosome or lysosome. But to really drill in on the structure, we notice that we have our V-type ATPases, we have lysosomal hydrolases, and intraluminal vesicles, which we discussed earlier, precluding receptor ligand interactions with cytosolic contents. To discuss the importance of this V-type ATPase, it is an ATPase, so it utilizes ATP to pump protons against its concentration gradient. So in reality, we would be seeing a ton of H plus accumulating in here, and we know that protons are associated with a decrease in pH, making this a very acidic environment. Okay, and this acidic environment is not very conducive to the actual structure of, say, a macromolecule like a protein, which is why we notice that when they're in this acidic environment, they are degraded, which in a process of autophagy that we may discuss in a later video. So to continue, we observe that we have our late endosome fusing with an endosome or a lysosome. They are synonymous with each other, similar to the process that we observed over here where we had our endocytosis vesicles early on fusing into an early endosome. However, here we have two fairly acidic endolysosomes, late endosomes, into a larger structure. And this larger structure, once again, is going to be rich in V-type ATPases and hydrolases at a pH of about 3 or 5. And there's a lot of hydrolases. Okay, so these could be nucleases, proteases, anything to essentially degrade and break down macromolecules. And it should be noted that this process isn't as harsh as it seems, even though it's a harsh environment, and the outcome of these macromolecules in terms of their structure is not the same as when it entered. That said, it should be noticed that products actually result from these hydrolytic processes necessary for, say, our cells to function. For example, say that we are currently existing in an energy deficient state. We are going to take the proteins, the functional proteins in our cells, then we are going to degrade them, say by adding ubiquitin, transporting them into a lysosome, and degrading them into free amino acids that can be used as fuel or building blocks for other more critical in the moment structures. Okay, so real quickly to summarize what we've discussed in this video, we begin with, say, receptor-mediated endocytosis, signaling to endocytosis, so to invaginate in the cell membrane, resulting in the formation of a vesicle that was initially clathrin-coated, but the clathrin coat sheds immediately, typically within seconds, resulting in the formation of an early endosome upon fusion with other endocytosis vesicles. Okay, we then move on to functions specific to an early endosome, which can involve recycling, transcytosis, TGN exchange, or simply maturation. And I should note that contents that by default undergo maturation lack a signal sequence that would otherwise redirect it to either one of these other processes. However, to move on, we have TGN transport that occurs throughout the entire maturation process, continually pumping, say, hydrolases and V-type ATPases into this endosome, making the environment increasingly acidic and increasing the concentration of these hydrolases, these hydrolytic enzymes, that only function at a low pH. They do function at physiological pH, but however to a nominal degree. So to move on, we have the formation of a multivesicular body, a late endosome, upon going a gradual loss in its tubules, vacuolar changes, changes in the actual Rab proteins and their associated Rab effectors, and movement toward the nucleus. Recall that the wrap proteins are responsible for the actual function of the endosome, the organelle, as well as its location. And finally, we have the culminating event, the fusion of a late endosome with a lysosome, resulting in a very acidic environment rich in lysosomal hydrolases, conducive to the actual degradation of many macromolecules. So that's really all I wanted to discuss in this video, and I will likely continue to post molecular bio lectures where I notice there to be gaps in online content. So thank you for watching and have a great day.